Hello, my name is Delia Vella, and I'm going to be presenting on viruses. So our agenda for today is that we're going to be seeing what viruses are. So we're going to see what are viruses versus cells, bacteriophages, viral anatomy, the lytic cycle, and the lysogenic cycle, and what are its differences. So first off, what are viruses? Viruses are very small infectious agents that can only replicate inside of the cells of the living organisms. Viruses versus cells. Viruses are extremely small, smaller than the eukaryotic cells, which are animal and/or plant cells. Viruses are not considered cells because they need a host to survive, and they do not have all the components that cells have, such as a cell membrane, cytoplasm, ribosomes, and DNA. So what are bacteriophages? So bacteriophages are basically a virus that infects bacteria. They can also just be called phages. So the viral anatomy is pretty much made of a virus is made up of two parts. And these are the main parts, which are nucleic acids, which are DNA or RNA, and a capsid, which is a protein coat. And some have a viral envelope, and others have like tail fibers and a sheath to be able to latch on to a cell. And then there's two different types of infection strategies for viruses. One is the lytic cycle, which makes new phages and results in the death of the host, which is to your left. And the other one is a lysogenic cycle, which replicates the phage DNA without destroying the whole cell, destroying the host cell, which is to your right. So let's go through the lytic cycle. So the lytic cycle, so it begins with the phage DNA. The phage attaches to a host cell and injects its DNA. Then the phage DNA circularizes, so it can either go to the lytic cycle or lysogenic cycle. So we're going to take the lytic cycle. So in the lytic cycle, the new phage DNA and proteins are synthesized and assembled into phages inside of the cell, and then the cell lysis releases the phages in destroying the cell, and then it creates a new phage and it spreads out. And in the lytic cycle, usually an example of this could be a common cold where you can start getting symptoms a couple of days after. And there's another type of infection strategy, which is the lysogenic cycle. So it still starts with the phage attaching to the host cell and injects its DNA, and then if phage DNA circularizes, and then it takes another route, which is the phage DNA integrates into the bacterial chromosome, becoming a prophage. So pretty much it mixes and combines into with the DNA of the host cell. And then what happens is that the bacterium reproduces normally, so copying the prophage and transforming it into daughter cells. So whenever the cell divides, it pretty much half of its DNA goes, and then after a daughter cell with prophage occurs, and then occasionally a prophage exists a bacterial chromosome, initially in a lytic cycle. So at the end, the lysogenic cycle will end up turning into the lytic cycle. And an example of a type of virus that is a HIV virus, where it can be dormant up to 10 years. And